Yeah, welcome to our third and final Simons lecture in applied mathematics. It's my great pleasure to introduce Professor Ray Goldstein from the University of Cambridge. So, as you have heard, if you were here the last 2 days, so Ray is chair of Alan Turing Professor of Complex Physical Systems at Delft. And his group is doing pioneering work at the interface of applied mathematics, experimental biophysics, and biology. But Ray also has a very broad range of interests beyond biological problems. And if you have seen ever a stalactite, so you can ask Ray about basically how it grows, or if you are interested in topological transformations of minimal surfaces, so I highly recommend to go to Ray's page, where you can find beautiful videos of Möbius-type soap films that undergo various transitions. And yeah, and as some of you may also know, so I probably wouldn't be here if it were not for Ray. So I was fortunate enough that about 15 years ago, a little bit more, Ray accepted me into his group as a postdoc, and I had an amazing time there. So yeah, so here that was hugely influential, basically both on my career trajectory, the type of work I do, but also I think life more generally. And yeah, so he has taught me how to identify interesting problems, how to approach them, and in particular also how to present them to the broader audience. Although, I mean, if we try to live up to his standards, but we'll never get there. And you know what I'm talking about if you were here for the last 2 lectures. So today's talk is about decision making without a brain. So I assume it's not about politics, but it's actually about biological systems. So Ray, yeah, very much look forward to you.
Thank you. I'm not sure how I can top that. Okay, so thank you, Jorn, very much for the introduction, and thank you all for sticking with me for these 3 lectures. So today I want to talk about something that's rather different than the last 2 lectures, just to put things in perspective. The first lecture was general introduction to multicellularity, a discussion about synchronization of these wonderful appendages called cilia, which power the locomotion of microorganisms and move fluid around all over our bodies. The second was really more about the architecture, the structure of multicellular organisms. But today is what I think is the entree into a whole set of questions that's really very important about the thinking of microorganisms and how organisms, for instance, that don't have a central nervous system can nevertheless respond to their environment in ways that look to us as if the collective behavior of all the cells involved must involve communication, but somehow it can't because there's no central nervous system. So that's the kind of mystery that I want to address today, and I'm going to do it in the context of a phenomenon called phototaxis. So the organisms I've been describing are all photosynthetic algae. They need the light to do photosynthesis. They have evolved to swim toward the light, as you see here, and it's a wonderful type of response, a kind of decision making. When you turn a light on, they turn and they swim to it, and the question basically is, how do they do that? So I'm going to use the power of this lineage of organisms that I've been telling you about so far, that spans from a unicellular organism all the way up to a highly multicellular one, to understand if there's anything common between the uni and multicellular organisms as far as the way that they do this process. And so I'm going to use 3 of these organisms. As you see here, on the left is Chlamydomonas, a single-cell organism, 10 microns across. If you happen not to have been here before, remember your hair is 100 microns across, so that's a reference scale. As 2 of these beautiful eukaryotic flagella, it swims through the fluid environment with a breaststroke motion. And it's the building block of many other organisms. One example here is Gonium. It's a 16-cell organism, each cell of which is conceptually like Chlamydomonas. It just so happens that the cells on the periphery have their flagella beating like this, and the cells in the center have their flagella beating like Chlamydomonas. So it's sort of a hybrid of 2 different swimming modalities. And then here's Volvox, which I've spent a lot of time talking about, that is 1,000 cells on a spherical extracellular matrix with a very highly organized beat pattern, as I will tell you. And we're going to try to study these by experimental and theoretical methods to understand how they swim toward the light and see if there's anything common. So the first thing to say is they all do have one obvious common feature in the way that they move through their fluid environment, and that is that they spin. So each of them has an axis around which they spin in a very graceful manner as they move. So Chlamydomonas indeed has these two flagella that are in this beat, this breaststroke beat. But if you looked at it from this side, you would discover there's a slight twist in the way it beats. And that makes it spin around this axis, this central body fixed axis. Because of the separation between the way the peripheral cells in gonium beat and the central ones, this object also spins around an axis coming out toward you. So the central cells pull it toward you, and the peripheral ones make it spin. Now in Volvox, it's a little more subtle. There is an anterior-posterior axis here, which is actually pretty much the axis along which all that inversion dynamics I showed yesterday takes place. But the cell position and orientation is such that the flagella are beating parallel to each other, roughly from the North Pole to the South Pole, but with a slight east-west tilt. And that makes the whole thing spin around that axis. So this is just the way nature has made them. And that's the important ingredient number one in all of these organisms as far as the way they sense light is that they spin. And you can have a long discussion about why they spin, but for the moment I'll just accept that they do. Now, this issue about why they spin and what advantage it might have is actually something that's been around a long time. And a few years ago, I was compelled internally to write an article, actually in Physics Today, to kind of exhort everybody to read the old literature, because you can discover all sorts of interesting motivations in very old papers and interesting observations about phenomena. And speaking of that, there's a beautiful paper from 1901 by Jennings, which is entitled On the Significance of the Spiral Swimming of Organisms. So Jennings was a very brilliant biologist. He was not a mathematician. And he was struck when looking through the microscope at the fact that so many organisms that he looked at, not just these kinds of green algae, would swim in a helical fashion. And he tried to understand why they might do that. So here's an organism elongated that would swim in a helix. And he said, for instance, the device of revolving around an axis compensates for any tendency to deviate from a straight course, as if basically it sort of irons out or averages out the fluctuations that might be occurring. But one of the things that he noticed, which is very important, is that if there's any spot that you locate on the organism, some organelle, some colored region, as it goes along this helix, that object on painted dot, let's say, has the same relationship to the inside or outside of the helix all the way. So if the dot is facing outside at some point in the helical motion, it's always facing on the outside. And likewise, if it's facing to the inside, it's always facing on the inside. It's not obvious that that should be the case, but it's true. And then he made another really interesting observation, which is that when you have an organism whose appendages are such that it could rotate around two orthogonal axes, so suppose it has two flagella, and by beating one, it rotates around one, and by beating another one, it could rotate around the other, that if it only actuates one, it goes in a circle. But if it actuates both, it goes in a helix. And that's also not an obvious thing. And we were puzzling about this at some point. And then I went to the source of all knowledge in mathematics, which is stack exchange. and someone put the following question. I want to show that a rigid body with two components of angular velocity vector and one of linear velocity, in the absence of external forces, has helical trajectories. So this is my homework assignment to you. And I'll tell you that one way to solve this problem is to find a connection between the Frenet-Serre equations that would describe a helix and the equations of motion of angular variation of a triad of vectors on a body. We'll get to that in a bit. So that's point number one, that these organisms have this ability to move in helical trajectories. And point number two is that each of them has a kind of primitive eye. It's not an image-forming system like we have. If anything, you could make an analogy to a camera by saying it's got a single pixel. And that photosensor can be seen, in a sense, if you have a good microscope with good color recognition, by looking, say, at the north pole, called the anterior pole of Volvox. Here are the somatic cells moving away down the sphere. And these red dots are not the photosensor itself, but they're actually a protein layer behind the photosensor that shows up red, and they're called the eye spot. So there's the photosensor and the eye spot. And the eye spot is a very important development in biology in the following way. Here's a kind of caricature schematic of the way light interacts with a Chlamydomonas cell in the case of the wild type organism, meaning the one you find well behaved in nature. So let the eye spot be red, let the photosensor be blue, it's on the outside. And if light comes from the right, it actually passes through the photosensor, and the structure of this eye spot is actually very non-trivial. It's made of protein globules in a regular kind of lattice-like structure in such a way that it acts as a mirror at these wavelengths, and it reflects the light back. And so the light does a double pass through the photosensor out the front. That means that if the cell were in the opposite orientation with respect to the light, so the light is from behind, it also acts like a mirror, and there's no signal received. So the photosensor apparatus with the eye spot is directional. It only cares about light from the forward direction, not from the behind. And that's important, as I will show you, in all of these systems. And the way to see that that's important is to look at mutants. This is the standard way in biology. So there's a class of mutants that's missing the eye spot. So there's no mirror-like structure behind the photosensor. So if light comes from the front, yes, there'll be a single pass through the photosensor. It'll get a signal. And if light comes from behind, you might think, well, it's also going to get a signal. And so it would be pretty much confused, because if it has the ability to turn toward the light, it'll alternate between going one way and the other. It turns out it does go a unique direction. It's just the wrong one. And the reason that was discovered by Ueki et al. Is that the cell body acts as a lens, because the index of refraction of the medium on the inside is higher by about 10% than the outside. And so the light is focused to be more intense in the opposite direction, and the cell says, well, that's where I want to go. But you can already see there's an issue, because it's going to get one signal like this and another signal like that if it's spinning, and it has to somehow make a decision. So keep that in mind. That's the two things. Spinning and directionality of the eye spot are the crucial ingredients in phototaxis, as we will see. So again, the way we're going to study this experimentally is to use the technology from in vitro fertilization or electrophysiology, where we have these glass capillaries, as you see here, that are necked down to hold something as small as chlamydomonas, as intermediate as gonium, or as large as volvox, and we're going to interrogate these under the microscope to watch what they do. And the typical way we do this is we have a little microfluidic chamber, and the capillary comes in like this. We can rotate the cell, as I mentioned before. And by holding the cell, it's much easier to figure out what the flagella are doing than if it's freely swimming, because it's hard to focus on them as they're moving so fast. And so then, instead of the cell spinning and experiencing what is basically a periodic signal because of this directionality in the presence of the sun, we're going to turn a light on and off at some adjustable frequency, mimicking what it experiences as it turns. And we'll use that to understand the frequency-dependent response of the photosystem. We're also able to track things in 3 dimensions. So imagine a chamber like this with an inner chamber where the cells swim. With 2 high-resolution cameras looking in orthogonal directions, we can locate the x, y, and y, z coordinates of the swimming cell and reconstruct the 3-dimensional trajectory and study how it actually swims. When you do this, you want not to confuse the organisms with the light you're using to see them. So we end up using red light. The problem is red light heats things up, and if you're not careful, you end up with thermal convection inside your chamber, and everything doesn't work. So you have to have a complicated heat bath to make sure that doesn't create things. But once you've done that, then you can look at the free-swimming organism nicely. So I'm going to go in reverse order. We're going to first historically go back many years where we studied volvox, then we're going to do gonium, and then we're going to do chlamydomonas, and then we're going to see what we learned. So here's the way we did it many years ago with Knut Drescher as in the lead, my then PhD student who's now a professor in Basel. So we hold the organism like this. We reorient it so that we're illuminating the front of the colony. And as I say, the fluid flow is driven primarily north pole, south pole along the organism. And this is an old and grainy video, but it's very important to show you what happens. So we have microspheres in the system, so we can track the fluid flow around the organism. And here the light is off, and when I play the video, you see them streaming by here. So there's a flow like that being driven by the flagella, and as soon as the light is on, the speed of flow initially and rapidly drops, and then it recovers over the course of a few seconds. So this is a kind of adaptive response. It's like what happens if you go into a room where there's a bright light, or you go outside, and you have a shock response to the bright light, things change, but eventually you get used to it. Or if you sense a strong smell, you have a shock response and then you get used to it. So we want to quantify this, and by looking at the the motion of these microspheres, we can get a vector field of velocity all around the sphere, and we can measure, say, in some units, the fluid flow speed as a function of time when we go from the light being off to the light being on. And this is what you see. You can call it a drop in recovery response. So it's characterized by two time scales, a short one that's measured in tens of milliseconds and a long one that's measured in seconds. Now, when you look at something like this, you might think that this is some kind of complicated nonlinear dynamics analogous to what is responsible for electrical impulse propagation along neurons, if you know things like that, or oscillations in your heart. But it's actually a perfectly linear response. And it can be described purely phenomenologically by a perfectly linear pair of equations, which I show here. So let's suppose that the amplitude of this drop is called P for the photoresponse, and let the signal be S, which in this case is basically 0, 1. And the idea is that there are two time constants here. One is tau response, which is short, and one is tau sub adaptation, which is long. Now, we're going to have the signal and the photoresponse and something called H, which literally means hidden. It's the hidden dynamics of adaptation, which we don't understand, but we're going to characterize it essentially the way two capacitors talk to each other by charging and discharging. So imagine you start with no signal, no H, and no P, and you suddenly turn the signal on to 1. Well, if H is 0, and I'm at an early timescale before H wakes up, meaning a timescale less than tau A, then all that happens is P tries to become S. So this is just the equation of a capacitor charging on a timescale tau R. So that's this drop here on a timescale that's tau R. But on a longer timescale, H wakes up, and H says, no, I want to be like S. And as H becomes equal to S, P is now turned around and trying to chase something which is going to 0. So it drops and it recovers, and those are the two timescales. It's that simple. But it's then ready for the next event because it's adapted. And so the picture you might have in this kind of smeared out world in which I'm not resolving what every individual flagellum is doing is there's a fluid flow over the surface, let's call it U, and there was a U naught, and it's just been corrected a bit by this P, which is time dependent. So that's the simplest phenomenological model that you could cook up that would show this kind of adaptive behavior. Now, the interesting thing is that I can Fourier transform that pair of equations and ask what would be the response of the system if I turned the light on and off at some frequency omega sub S for signal. So, of course, the organism rotates at some characteristic frequency, but I'm free to adjust this frequency to be whatever I want. And what you discover is that it's actually a bandpass filter in the sense that it has this peaked response function, note the log scale, here's the mathematical formulation, in which it doesn't respond at low frequencies. It doesn't respond at high frequencies, but there's a middle frequency, which is basically 1 over the geometric mean of tau R and tau A, where there's a strong response. So that's point number 1. It's a bandpass filter, and it makes sense biologically. If I'm an organism and I'm underwater and I'm looking at the sun and I want to go that direction, I don't want to be distracted by high-frequency fluctuations of the surface of the water that are creating fluctuations in the light intensity which are very fast compared to my own motion. And maybe I don't want to really care about the slow dynamics of clouds coming by as well. I want to be sort of tuned somehow. Now, the interesting thing is, remember, perhaps if you were in my talk yesterday, that when Volvox colony is first hatched from the mother, it's very small, and then it grows to be very large without any cell division over the course of a day or so. Now, if you measure the spinning frequency, the rotational frequency of the colony as a function of size, or equivalently age, it's a very strongly decreasing function of size. Basically, the rotational drag on a sphere goes like r cubed. So as you increase r, there's just a huge increase in the drag, and so it slows down. And it just so happens that the cells that are really good at phototaxis, they rapidly turn toward the light, are those whose size range is in the purple bar here. So they're young, because they're small. Now that purple region corresponds precisely to the peak of this bandpass filter. So their rotational frequency is tuned to the response of the photosystem to the light. So what does that mean? Well, it means that if I'm a Volvox colony and let's say the light is coming from over there, as it is with the projector, and I rotate, a little patch of the surface rotates into the light, that it will immediately drop its fluid velocity created by the flagella, but it will recover on some time scale. And if the time it takes to recover corresponds roughly to half an orbital period, then the net effect will always be that the dark side beats faster than the light side, and you'll turn. So the tuning makes sense if I want to turn effectively. And you can mathematize this in the following way. There's a wonderful result due to Stone and Samuel up the street at that small liberal arts college, based on what's called the reciprocal theorem in fluid dynamics, which allows you to express the turning velocity, let's say, of a sphere, whose surface is endowed with a slip velocity, u, by crossing that with the normal to the sphere and integrating over the whole surface. That will give you the angular turning rate of this sphere. Now, it's pretty clear if u is a highly symmetric, axisymmetric distribution of velocity, this would be 0. You have to have some asymmetry. I just told you how you'll get an asymmetry. You'll have the dark side beats faster than the light side. So you can endow a sphere with the same pH dynamics that I showed you before and let it respond to the light and see what happens. And in the frame of reference of the sphere, the light just evolves by solid body rotation. So if you do this and you color code a sphere such that blue represents strong beating and red represents this downregulated beating, then if you start out with it, say, orthogonal to the light, first thing it does is downregulate the beating here. That makes it turn. But the more it turns, the more the light is continuously illuminating it. And by the adaptation method, it recovers and faces the light, beating as strong as it did originally. So that's a fixed point of the dynamics. And the important lesson here, which is what I want to stress when it comes to understanding the evolution of multicellularity, is there need not be any discussion between the 1,000 cells on the surface. All that has to happen is they have the right response to the light wherever they are on the surface. If they're doing the same thing, this dynamics will make them line up. We think that there is coordination between them because we see everybody doing the same thing, but they're not responding to each other. They're responding to the signal at the front. And the analogy I like to give is if you go to a church on a Sunday morning and suppose the door to the inside has a glass in it and you look through and you watch and every once in a while everybody stands up and then they sing and then they sit down and then they do it again and you think, oh, they're talking to each other and saying it's time to stand up. But no, all they're doing is responding to the person at the front with the right response. So the person at the front is the sun. And you can test this idea because it turns out that the flagellar beating dynamics is very insensitive to the viscosity of the fluid, but the rotational frequency is extremely sensitive to the viscosity of the fluid. So you can add polymers to the solution, which increases the viscosity and slows down the rotational frequency, and have an assay for how well they do phototaxis. And it drops like a rock as you detune the system. And it's fully consistent with this simple picture that I showed. So this shows one of the first steps toward functional multicellular organisms is just having evolved the right kind of response to an environmental cue without having to have any nervous system. Okay, so now let's ask what happens if I go to a different organism, and here's gonium again, a beautiful micrograph of gonium. You can see the extracellular matrix that attaches one cell to the next. The flagella got a bit damaged in making this picture, but you can see some remnants of them here. It's been studied for a long time. It can be a 4-cell organism, an 8-cell organism, but in healthy young cultures, it is 16 cells, just like you see here. So you would like to basically do the same kind of experiment and see whether the same result holds. So here again is the swimming of gonium. It's this beautiful, almost flat plate that forms this wonderful rotational helical motion. And if you turn a light on and off alternately on two sides of a microfluidic chamber, you can see how nicely they respond to the light. This is sped up by 10 times. They go over here. They just turn around like plates navigating through the world. So it's very phototactically responsive and quite nice to study. Okay, so the experiment is pretty much the same. You hold it on a micropipette in this geometry. You shine a light from below, and you have microspheres in the solution, and you can track the fluid velocity in this geometry. And what you see is when you turn the light on, there's a drop on the side that's illuminated, but not much on the other side. And eventually it recovers. And if you quantify it here, you see this drop in recovery response on the illuminated side, but not on the shadow side. So it's a little bit more subtle how it actually does the turning. It turns out if you make a kind of force diagram of what the flagella are doing, the ones in the periphery are not exactly beating in the plane. They're slightly tilted. And so when you suppress the beating of some portion of that, that leads to a torque that turns the organism toward the light. But you can now study many, many instances of this effect and measure the adaptation timescale and the response timescale. The red line on this log-log plot is a line of equality. And what you see is there's a lot of noise, but the adaptation timescale is always pretty much larger than the response timescale. So there's this separation again. So that shows that gonium is consistent with this picture. And now let's turn a little to Chlamydomonas and see what we can learn. Now, Chlamydomonas has been studied for a long, long time in this sense. And actually, the original important people are, again, this pair of German biologists, Ruffer and Milch, who in the early 1990s and thereafter studied with high-speed cinematography what was going on. And here's a nice diagram from their paper. This looks like an organism that has only one flagellum, but it's actually just the normal organism with two, where they're showing graphically what one does and what the other one does for your convenience. And the interesting thing here is that there's a built-in asymmetry between the two flagella. So if I start in the dark and I turn the light on, I will discover that the transflagellum, the one that's furthest from the eye spot, will increase the amplitude of its beating. So the dashed line is sort of the normal envelope of dark beating. So the transflagellum will increase in the presence of light, and when I turn the light off, it decreases. And then we'll come back. Whereas the cis has the response that when I turn the light on, it decreases the beating, and the other one increases. So the two flagella are not identical, and their response to internal biochemical changes are distinct. And without that distinction, this doesn't work. And now, if you fast forward a little bit to later work by Joseph Saranac and Foster, who first tried to understand the frequency-dependent response of the behavior. They did the experiment analogous to what we did with Volvox. They didn't have an imaging system. They had a quadrant photodiode, which is 4 pixels. And when you illuminate, when you project essentially the image of this organism onto such a thing, from the voltage difference that you get in the 4 quadrants, you can get a sense of where the flagella are. And if you look at the frequency-dependent response, the amplitude and the phase, you see again this bandpass filter behavior with a frequency peak that's pretty much at the rotational frequency of the organism. And there's even a measurable phase shift. So it's a kind of coarse-grained measurement. There was no detailed waveform analysis, but they clearly show that there's this bandpass filter response. So we wanted to go beyond this and try to understand the detailed waveforms. So the picture that emerges is really an interesting one. It's that if the light is coming from the blue arrow direction, that when the eye spot rotates into the light, the cisflagellum down regulates its beating, the transflagellum goes up, but half a spinning period later, if they did the same thing, they would undo what they just did with respect to turning, and so it has to be the opposite response, so they keep going in the same direction. So there's this intricate dance of alternating responses that apparently leads to motion toward the light. So that was the state of things back then. So our approach to this problem is we have high-speed cameras. We have the ability to digitize these waveforms. We have the ability to think about the theoretical implications of this. How would we do this? So you see a video up there showing you this high-speed beating. This, again, is beating at about 50 or 60 cycles per second. So we're using high-speed cameras of 2,000 or 3,000 frames per second to get really accurate waveforms. And the approach we're going to take is that we would like to be able to estimate from the observed waveforms the forces and torques that act on this organism to turn it. So remember, I told you that the two flagella naturally beat slightly out of plane, even when there's no photostimulation. That leads to spinning around an axis I'll call E3. There are two other axes. Let's call E1, the one that comes out this way between the two flagella, and there's E2. And the transient asymmetries between the flagella will lead to torques that spin me around E1, even while I'm spinning around E3, like Jennings was talking about. So to estimate those, we're going to use a result in fluid mechanics, which is an approximate description of the forces exerted on moving slender objects. It's literally called slender body theory, or resistive force theory is the simplification of it. So the idea is a quasi-local description of the dynamics. So the force acting on the fluid, let's say, as a function of arc length and time is related to the velocity dr dt of the filament at that particular point and time via a tensor which has two coefficients that represents the drag coefficients for motion perpendicular to the long filament and parallel to them. And there's a beautiful result in fluid mechanics that basically comes from looking at ellipsoidals, objects where you have exact solutions, that the drag coefficients for perpendicular and parallel motion are roughly in the ratio of 2 to 1. And they look like 4 pi times the viscosity divided by the log of the aspect ratio, length divided by radius of the filament. So it's basically a local relationship between force and velocity. So if I have a digitized waveform, I can calculate the forces. So if you have, of course, steady beating without any light signal, you can average for thousands of beats and get beautifully smooth data. And this is what you get. Here are the forces on the left, coming from the left and right flagellum. This is the propulsive force in the direction of motion. As a function of time in units of the beat period, it's a nice sinusoidal behavior, because what happens here is there's a power stroke where the flagella are basically straight, pivoting down like this, and then there's a recovery stroke where they go like this. And the difference in effective drag between the power and recovery allows forward motion. If I were just doing this at 0 Reynolds number, I wouldn't get anywhere. That time reversal motion does not lead to net translation. So there's a large fluctuation in this. The mean is actually relatively small. You can also then say, well, if I'm going to turn the body, I could estimate the torque around, say, the body center. By the way, the scale of forces here is piconewtons. That's the natural scale for here. And the torques would be forces times distances, piconewton microns. And again, these are, of course, antisymmetric roughly because the two flagella are turning in opposite ways. And the scale of them is hundreds of piconewton microns. So that's the level of quantification we can do, but that's just for steady beating. So now there's a question here. For Volvox, I told you there was this wonderful little adaptive dynamical model, and we could think of it in terms of the fluid flow over the surface of the sphere. And that makes sense. I've got thousands of flagella from a distance, or not even very far. It's clearly smeared out into something like an effective slip velocity over the surface. And maybe when it comes to gonium, I could get away with that because there's still a fair number of flagella, or I could think about the forces of those individual flagella. But it would be pretty ridiculous to sit here and say, I'm going to abstract 2 flagella coming out of this sphere of climbing the bonus as a slip velocity. That's just too much of a coarse graining picture. On the other hand, just think about it as rigid body motion. If there are torques coming from these flagella, then the angular velocity of the object is the fundamental thing I'm interested in. So we're going to investigate a hypothesis where the dynamical system we had before, which referred, say, to the fluid velocity, is actually applied to the angular rotation velocity of the organism. And indeed, if there's a transient flagellar asymmetry, there's a transient angular velocity. So let's try this. But I want you to appreciate that if you actually looked at, say, the torque coming from one flagellum or the other flagellum, well, they're each oscillating at 50, 60 hertz. And we're talking about a long time slow modulation of that beating that comes from this transient response. So we're not trying to describe the up and down oscillations at 60 hertz. We're trying to understand the sort of running average of these dynamics. And that's the timescale in which this will hold. And then if you think about this dynamics, pretty much the maximum response that you can get from this system is set, the maximum angular velocity is set by the signal. And so you can measure the signal in units of the angular velocity maximum that you would have. So that's a parameter of the problem. How much net angular velocity could you get in the extreme? Okay, now it's not easy to do this experiment because whereas you can just sit patiently and let 1,000 beats go by for the steady beating, here we're looking at a transient dynamics. So you have to try to average over transients, and you can do that. My amazing postdoc, Kyriakos Leptos, pioneered these experiments, and this is what you see. So it's noisy, but there's a clear drop in recovery response, which you can convert to a typical kind of torque on the system or a typical kind of angular velocity measured in a few radians per second. Now, this is the bandpass filter on a linear scale, not a log scale, and it's very much consistent with the measurements we make. So we're seeing the bandpass response just like Joseph Saranac and Foster, but measured in terms of the real torque on the system, or the real angular velocity. and even the phase matches pretty well. But now there's a problem, at least conceptually. The dynamics, as I set them up here, has the signal, or the angular velocity, responding to the signal. But what Ruffer and Nilch told us was that actually it's the derivative of the signal that matters. Because when you step up the light, you get one behavior of one flagellum. When you step it down, you get the other. That's not the signal, that's the derivative of the signal. So how could this possibly be a reasonable model for Chlamydia monas if it cares about the signal and not the derivative? And the answer is something you learned in nursery school, kindergarten, I don't know, which is that, 2 first-order equations is 1 second-order equation, right? And so if you just eliminate h from the problem, you'll get an equation for, a single equation for the angular velocity omega-1 in terms of actually s dot. So it's actually a damped harmonic oscillator responding to the derivative of the time signal. And what's really interesting about it is that, remember, the 2 time scales are very different. The response time is very short, the adaptation time is very long. So sitting in front of the highest derivative in the problem is the small parameter. So it's actually a kind of singular perturbation to get rid of the response time scale. But it really shows that actually this is the simplest kind of model that would show this antisymmetric response with respect to stepping up and stepping down, and that's very important. And you can see it here. I take a square pulse, and my response will be an up and down recovery when I go up, and it'll be down and up when I go down. Okay, so here's my slide to show you that I am capable of doing mathematics. And so the first thing is, how do you turn this into a theory of motion? And the answer is, first, you pick up your favorite textbook on classical mechanics, which is surely the one by Goldstein, and no relation. And you look up the part on Euler angles. How do I describe an object in space that's got some structure to it? Well, I need to, say, have one angle down from the North Pole, one angle down around here, and one angle that tells me how much I turned around. So those are the 3 Euler angles, phi, theta, and psi, that I would use. And you can view this 2 ways. You can say, maybe I know the angular velocities omega-1, omega-2, omega-3, and I'll find phi. Or maybe it's the other way around. I know what the angles are doing, and I want to find the angular velocity. In this case, there's an omega-3, and there's a transient omega-1, but there's no omega-2. So this is the simplified version of the problem, in which I have only those 2. And then I have my dynamics for the photoresponse. However, the thing is that there may be an intrinsic, as Jennings taught us, an intrinsic difference in the torques produced by the 2 flagella, even in the absence of a photo signal, and that would lead to helical motion. So there are 2 parts, possibly, to my angular velocity. There's an intrinsic part that gives a helix, and there's a photoresponse part. And actually, Chlamydia monas does swim in a gentle helix, so you could put that in if you want. Okay, and then my signal is actually the maximum possible one I could get. And then it's the projection of the light on the eye spot, or the photosensor, times a Heaviside function of that, because I don't see the signal from behind. So I have this rather strange JHJ here, where J can be expressed completely in terms of the Euler angles, and some angle that describes where the eye spot is with respect to the 2 flagella. It can be distributed in various places. So it's a completely closed system, and the thing to do is actually to rescale it by the characteristic spinning speed, omega-3, and define a rescaled time t and rescaled s, h, and p, and you end up with a 5-dimensional dynamical system. So it's modular in the sense that this is all about geometry. This expresses what the Euler angles are doing in terms of the photoresponse. And then there's this rescaled version of my adaptive dynamics. And it's modular in the sense that if you came along and said, no, I have a better theory of the photoresponse, you just take this part out, swap it in, and the rest is unchanged. So it's completely closed. And the question is, does it work? And we can measure all the parameters in the problem. So let me just show you it works in the sense that you slap this on a computer, you write a little MATLAB code to do this, and you start with a sphere, let's say, facing nearly opposite the light. So there's a tiny little signal that the eye spot gets as it spins around. And when it gets those signals, it will trigger a photoresponse P, which will trigger turns, and the system will actually completely turn around and swim toward the light. And that's a fixed point of the dynamics. And here's the signal JHJ. It grows to be a maximum when the cell is orthogonal to the light, because now it's spinning with great projection on that, and then it dies away to zero as it lines up. So that's good, but it doesn't really explain how it works, or quantitatively what's going on. So here's a way to think about it that again exploits a separation of timescales, because the spinning motion is pretty fast compared to the slow turn that the object makes toward the light. So to get some intuition, you say, well, let's just freeze the Euler angle phi, which describes my orientation to the light, and just think about what's happening to the photoresponse at a given phi. So my signal for some particular orientation phi will just be some p star sine t, heaviside of sine t, a rectified sine wave. And from that perspective, this is just like the Volvox problem we studied in the first place. If I have light and I turn it on and off, and I have a sinusoidal forcing, the response of the system will be given by that bandpass filter. So I know, just from general physical principles, that my photoresponse will be proportional to the signal, and it will just have some gain function, which I'll call g of omega 3. That's my bandpass filter, and there'll be a phase shift pi, which I know analytically. There's a factor of a half, because that's the Fourier transform coefficient of a half-wave rectified sine wave, the lowest mode. So if you then unfreeze the angle, you end up with this very simple equation. d phi dt, the orientation toward the light, is just cosine phi, where the timescale, tau sub x for reorientation, is given by 1 over this function q, where q is related to the bandpass. So the timescale is 1 over the bandpass function. So if the bandpass has a peak, this timescale has a minimum. And if you plot it on this log linear scale, for different ratios of the response time and the adaptation time, you see a very strong minimum right about at the point where the rotational frequency of the organism times the adaptation time is of order 1. If that's 1, that's tuned, as I described to you earlier. So there are two things you see here. This represents, apart from some factors, this is the turning time toward the light. And it's minimized if you're tuned. So there's optimization in the sense that if you're tuned, you will turn the fastest toward the light. But it's a broad minimum. I mean, this is a log scale. So actually, there's also robustness in the sense that if I were to deviate my parameters from being precisely tuned, just think of the graph in Gonium, the tau A was all over the place. I will have to go really far away before I start suffering here. So there's both optimality and robustness, which I think any biological system ought to have. Okay, so you can test this. We have this three-dimensional tracking system. We have cells swimming in the chamber. We turn on the light, and we watch how a cell reorients and swims toward the light, and we can measure the orientation angle with respect to the light. And if you just shift by a factor of pi over 2, you can see this beautiful evolution that corresponds to alignment. Those are the circles. And the solution of this equation matches it beautifully. Right? So basically, it works. And in fact, if you estimate this time constant from the resistive force theory argument that I gave you, these different versions of it, it pretty much matches what you see experimentally. So it seems like it's a done story, but you can tell from the way I'm setting it up, there's a problem. And the problem is the following. And this was really a very interesting example of reconciling the way biologists and mathematicians and physicists think about things. So Schaller et al., who wrote this very influential paper in 1997, said the following about phototaxis and the orientation of the eye spot. So there are several different species out there, some of which have a dominant cis-flagellum, some of them have a dominant trans-flagellum in the absence of a photocue. And they all have a left-hand spinning, but some of them move toward the light, positive phototaxis. Some of them move away from the light, negative phototaxis, depending on light intensity. And they made a little table that basically, where is the eye spot? Where is the photosensor? Think of Jennings. That's a painted dot on my surface. Where is it as it goes around this helical motion? Is it on the outside or is it on the inside? And there are all these different combinations that you can see. And one of the things they observed is that it appears that the position of the eye spot is such, and the spinning is such, that when it actually lines up with the light, remember there's a bit of a helical motion, there's a slight tilt to the organism, the eye spot is actually in the shade. So it's like saying the way I walk toward that wall is I go backwards, so my eyes don't see it. But that seems to be what's happening, right? So the end result is, of all of this turning is that I hide my eyes from the light. And that's how I go toward the light. And so the reason they rationalized it this way, they said if this state has been reached, that is, the signal that tells the organism not to turn anymore is not the now reached constancy of perceived light. That would be if it was still in the light, but which needs to be interpreted by the organism, but it is the absence of an internal light-induced signal. To translate it, if the alignment has the eye spot facing the light, but nearly orthogonal, there's a weak signal there, and somehow the cell has to say, that tiny little weak signal, that's exactly what I need, and I know I'm lined up, as opposed to, oh, there's no signal at all, then I know I'm lined up. And they said the latter makes more sense. I mean, it made sense to me, but is this really correct? So this is an issue about which of the flagella is dominant, even in the absence of a light signal. So we decided, let's just look at long-term beating dynamics of a bunch of cells using this ability to track the waveforms and estimate the torques on the two, and see what the probability distribution of basically average torque is over all these cells. So if you average over dozens of cells and make a histogram, it's a beautiful Gaussian, and it's pretty much exactly centered at 0. And you might say, fine, but some of the cells actually then have the cis flagellum dominant, and some of them have the trans flagellum dominant. And yet they all do, in this pool of organisms, they all do positive phototaxis.
Is that orientation relative to the spiral? Is it relative to the eye spot, or is it relative to the spiral of the helix? The difference between cis and trans?
Cis and trans is relative to the eye spot. So here's the eye spot, and this is trans, this is cis. Yeah. So this one is dominant, or this one's dominant, and that determines the helix. And the answer is the cells that respond to the light in a unique way, like positive phototaxis, have a distribution, some of which is this dominant and some of which is that dominant. So it completely shoots the argument of Schaller et al. But yet, you know, it seems plausible. So how do we reconcile this? The answer is they weren't thinking of an adaptive dynamics. They were thinking a signal is interpreted by its value, not its time history. And so the answer is if you have a steady signal on the photosensor, it doesn't really matter whether it's 0 or finite, the system will adapt to it and then not care about it anymore and not turn anymore. And so you can take our calculation and you can set up a situation where you have one cis dominance or trans dominance, and you can look at the light signal that you get as it lines up. And in the case of cis dominance, well, the signal eventually goes to 0. That's putting it in the shade. But in the trans dominant, it goes to a constant. But once it goes to a constant, there's no more turning because it's an adaptive dynamics. So that's the reconciliation of the two problems. The adaptive dynamics fixes this apparent contradiction. The other thing to say is that the eye spot is, yes, it's an object that's proportionally about this big, but there's a lot of jitter in the beating of the flagella. I showed you this in the first lecture. There's all this internal noise, and that rocks the position or the orientation of the cell. And actually, if you think about how much rocking goes on, it's way in excess of the size of the eye spot. So it just stochastically, it's sometimes above and sometimes below. So it would be pretty weird if the system were not robust to that. So that solves that. So this story then says the following. If I take all the information we have for Volvox, whose radius is something on the order of 200 microns, let's say, or 300, and I compile it and I ask what is the mean and the error in terms of the degree of tuning, it's 1 within experimental error. And now and that represents the two hemispheres of Volvox responding to the light. And now if I go back to gonium and I do the same averaging, it's also beautifully tuned, but now it's the two edges of gonium. And finally, now if I look at chlamydomonas and take all the data we have, it's also tuned within experimental error. But now it's the two flagella. So this is an evolutionarily conserved response over, 3 orders of magnitude in cell number, so 2 orders of magnitude in size. It's been conserved because it works. And again, it doesn't require any discussion among the cells. So I think that's a very interesting result, but it also raises some interesting issues from the point of view of if nothing else, sort of robotic control. That the nulling out of a periodic signal, it seems to be a robust way to navigate. It's like many electronic devices we use in the lab to get rid of extraneous signals by locking into a certain frequency. So it raises some interesting questions about robustness of this control strategy on many different levels. Okay, so all of that is a story about the response to one light. And a couple of years ago, I was asked to be on the thesis committee of a student of Raphaël Généré, who is a brilliant experimental biophysicist in Paris at the École Normale. And we got to talking about things, and both he and we had been thinking about, well, what happens if you give them two lights? How do they make a decision between them? So I'll tell you about the result of that just briefly. So the setup is chlamydomonas, just like before. Under the circumstances studied here, it's negatively phototactic, so it flees the light, but it doesn't really change much in the whole discussion. So the experiment, which was really carried out primarily by Shantanu Raikwar, involves having a petri dish, basically, or a thin microscope slide chamber, in which two blue lights are, that are collimated are illuminating the central region. They have adjustable intensities. Basically, think of one having intensity 1 and the other an intensity I'll call eta, and adjustable opening angle between them. So you can change the angle and change the relative intensity. And this is what is going on here. So suppose the light is coming from these two directions, V plus and V minus, and the cell is here with its body fixed axis along E3. As it spins during one half of the cycle, the eye spot is over here, the other half, it's over there. And there are these different regions of space such that depending on where you are, you may have the setup such that the eye spot sees both lights during a half turn and none during the other half turn, or one light in one half turn and one light in the other half turn. But nevertheless, they found an incredibly simple result, which they just wrote down by inspiration. They said, there's a vector along which these cells swim. So you have thousands of cells, you track them, there's a well-defined direction they all move in that is somewhere between these two lights. So it's not away from just one light, it's not away from the other, it's some compromised direction. And you have only two vectors, right? You've got V plus and V minus, and you've got one scalar, which is the relative intensity. And they said, well, it's probably this. You take a weighted average of the two light directions, normalize it, and then that tells you the swimming direction. Or if you turn it into the angle that they swim at with respect to the x-axis, tangent of phi star, the swimming direction, is tangent of delta, the opening angle, times an intensity-related function there. So we'll call that the tangent angle. And it's a brilliant summary of everything. And here's the data. It's really impressive. So here's phi star as a function of eta for different values of delta, and the lines go right through the data with, within experimental error. So this is a fact. And I remember when we heard this, the question was, is this actually something we could understand? And so we thought, well, what's the simplest generalization of the previous argument I gave you? Well, we found, with a slight shift from a cosine to a sine, before that d phi dt, the swimming direction, evolves toward the light by sine of phi. It previously was cosine, but I've changed the coordinates. So that was when the light was along the x-axis. So if the light is here and there, you probably would just guess that the generalization of the equation of motion is this. It's a weighted average of two contributions according to the two directions. So that would be our guess from our theoretical background of what the equation of motion is. And it's not obvious, but if you actually ask for the fixed point of this, it's that. So apparently, if you essentially average over the spinning and make the assumption that it's just choosing among the two lights by averaging and going forward, you get exactly the tangent law. So that's point number one. Point number two is that they then did another clever experiment, which was to start with one light on and have them all fleeing, quickly turn that off and turn the other one on, roughly right angles, and watch how it reorients in the new direction. And that's what you see here, red and then in blue. And they were actually able to track at different light intensities the angle that the trajectory makes with respect to the new direction as a function of time rescaled like we did before. And these different trajectories all kind of define a well-defined approach to zero. And if you look back at our harmonic oscillator forced by the function, you can basically realized that the parameters are such that you can, with alpha much greater than beta, that basically there's a dominant balance between this term and this term, which means my photoresponse is basically like the signal. And then if you put that back in the equation of motion, you get something in the red that pretty much characterizes this. But the most interesting thing that they found was when they went to the regime where the lights were nearly anti-parallel. And this is something that was suggested by our collaborator, Nir Gov, who was visiting on sabbatical, because he was interested in these kinds of problems, that there might be some bifurcation that happens somewhere. So here's a subset of trajectories when the lights are nearly anti-parallel. You can see some cells are swimming. These are noisy helices. They're swimming away from that light. Some cells are swimming away from that light, neither of which is consistent with the theory I just showed you, because the theory I just showed you said when the lights are equal intensity, they should go down the middle. That would give you a fixed point of 0. And there are some trajectories that could go down the middle. So some of them follow the geometric law. Some of them flee one light only. Some of them flee the other light. And some stochastically switch from fleeing one light to going down the geometric law and then fleeing another light and back. And this is not consistent with any of our theoretical analysis because everything we said would require them to go down the middle. So something's wrong in our hypothesis. And again, I can appeal to the variation in physical agility in the room or just proportions of body length and size. You know, we made the assumption that the eye spot was always on the equator. And if you just Google chlamydomonas eye spot and just look at the pictures that are on the web, you'll see that it could actually be all over the place. There's a distribution of positions. So the hypothesis is that actually the eye spot might be displaced in these different subpopulations away from the equator. And if you think about it, if it's just exactly on the equator and the lights are, say, perfectly anti-parallel, then slight deviation, if it were swimming toward one light, the slightest possible tilt will make it see the other light, and then it will go there or somehow reach a compromise. But if the eye spot is further forward, let's say, you have to rotate through a large angle before it finally sees the other light. So it can get locked into going the wrong way. So we said, can you measure the distribution of where the eye spots are? And it turns out there's a technique invented by the Japanese biophysicists some years ago in which you can. It acts like a mirror. It's just like when you see a cat's eyes or dog's eyes at night, they retroreflect the light. That eye spot's doing exactly the same thing. So you can measure where it is. But it's also clear that this is a stochastic process. So if we were to generalize our problem, it would be d phi dt is p sine t for the turning angle plus some noise. So it's a stochastic process. So they went and we did two things. We analyzed the statistics of the trajectories in little segments and came up with this three-peak probability distribution. Some is swimming away from one light, some swimming away from the other, some in the middle. And then here's the experimental distribution of eye spot displacements from the equator. It's a beautiful Gaussian, pretty much centered at 0, but with a width that is significant compared to the size of the eye spot. So there is a significant fraction of cells with the eye spot above the equator and those below the equator. And if you put this distribution into that stochastic version of the model that I described, you get out a PDF for the swimming trajectories, which looks just like the experiment. And you can then go in and say, oh, when the eye spot's in this direction, it corresponds to this peak, and when the eye spot is in this direction, it corresponds to the other peak. And in fact, there's a kind of bifurcation that does occur. If you sit at a fixed angle between the lights and you construct from this probability distribution an apparent free energy, and you just take minus the log of it, you can get a probability distribution that as a function of the displacement of the eye spot undergoes a kind of bifurcation from 1 minimum to 2. Okay, so the last thing I want to say is I want to go back to another aspect of this optical problem and just point out that there are some really interesting applied math problems to be found by looking at organisms in the marine world when you think about the fact that they can be not only photosynthetic, but they may also give off light. If you go to the right kind of beach at the right kind of time of the year at night, and you walk along or you just watch crashing waves, you'll see an eerie blue glow, which comes from organisms called dinoflagellates that are sensitive to shear and give off flashes of light. Just Google bioluminescence on the beach and you'll see that. And organisms that do that, for instance, are Pyrocystis lunula. This is a several hundred micron across dinoflagellate. It has a very complicated central structure consisting of light-generating organs that, through a complicated process of shear sensing in the cell membrane, triggers these pulses. There's also the fusiformis version of it, which is more like a axisymmetric cigar shape. And we started thinking, actually, that there are two interesting aspects of the optical properties of these cells. One is the photosynthetic problem of light coming into them. Remember I told you about the eyeless mutants and the cell body acting as a lens. Well, consider this as a lens. This is axisymmetric. This is not axisymmetric. What are the predictions we could make, or the analysis we can make, about the focusing of light to, say, photosynthetic centers in this cell, or the emission of light from outward? It's actually a really interesting and not particularly well explored aspect of optics because it's non-axisymmetric. So that's just a little bit of advertising. This is an interesting problem. We have a paper on it I won't be delving into, but I want to just talk about the simple problem that the Japanese who studied the eyeless mutant defined. So you just go to a ray tracing algorithm on the web and you set up a situation where you have a sphere whose index of refraction is 10% larger than what's on the outside. That's pretty much the regime we're in with Chlamydomonas. And you just ask yourself, what does the focusing of light look like in this system? And this is what you see. So as in any lens, there's a region at the top and bottom where the light is absent. And there actually is a kind of caustic at the edge of this because there's a non-monotonic relationship between the incoming angle of the light and the angle that you get over here. And so there's a diagram where you have to apply Snell's law in this kind of geometry. It looks like something out of Newton's optics. Probably is, actually, where you actually have to calculate all this. It's kind of fun. And if you then ask yourself, as a function of the angle that I might rotate a cell through in the presence of this kind of light intensity, so let's call the zero angle here, and I rotate the eye spot through this, well, in certain regions I get light from the front, and I just get a sinusoidal projection. But when I look in the lensed region, I'll get nothing, and then you actually get this very non-trivial, I call it like the Batman shape with the ears, this very non-trivial distribution of light, and these peaks represent the caustics, where there's a fold backward in the position of the light. So this is what the cell actually has to contend with when it's missing the eye spot. The integrated light intensity in this region is identical. You didn't lose any light rays. So the total amount of light integrated over this whole distribution is the same, obviously. But remember that Chlamydomonas is mostly responding to the time derivative of the signal. And so this signal is bunched up in a narrower region with much stronger time derivatives. And so when you put this into the theory of phototaxis, you discover that actually it's a decision-making process all over again. So if you had a population of cells responding to the light, those that were literally swimming toward the light, let's say coming from this direction, would continue to go. But if you were slightly displaced, you actually will see the stronger signal in the lensed case and go the wrong way. And so actually the ensemble of such trajectories for a real collection of cells randomly distributed would consist of a small number going one way and a large number going the other way. And they actually don't swim exactly toward the light. They actually swim at some funny angle, which is reminiscent of what we saw with the two light problem. I don't know if this is correct, but this is what the analysis says. So the next step, maybe some of you can help, is to just go in the lab and test whether this is the case for those mutants. But they're responding to the time derivative of the signal. This one has a much higher time derivative because of the lensing effect and goes the wrong way. Okay, so I'll stop there. I hope this gives you an idea of how, again, we can use the scientific method to try to unravel something about how these multicellular organisms operate and how it's connected to the unicellular one, and learn some lessons that might be relevant to understanding the transition from uni to multicellular before you have a central nervous system. Right, thank you.
Thank you for another wonderful lecture, Ray. There's time for a few questions, if there are any.
Or answers.
So one short question, one maybe longer question. So why would it swim away?
Well, it's getting confused, right? It's a mutant. It's got a problem. It's missing.
No, but I mean,
missing-
so all the ones that swim away are mutants.
Well,
I'm
so
not,
what's happening here-
I don't mean in this context. I mean, earlier you were saying.
So many of the cells have the feature that at low light intensities, they swim toward the light, and at higher light intensities, they swim away because the photosynthetic reactions produce what are called reactive oxidative species that are very corrosive, basically. And so this is a protection mechanism.
Okay. Yeah. And so, I mean, at bottom, there's this very simple mechanism where you have a motor and a detector, and magic happens. Do you see this in other, other situations?
So many organisms responding not only to light but to chemical signals, gradients of chemical concentration, swim in helical patterns and display something analogous to this. So sea urchin sperm, for example, navigate along an odor plume from the egg by moving in helical trajectories and sampling this plume and staying along the edge, and again doing something almost identically mathematically of changing the properties of the helix so as to go toward the source. So in chemotaxis and in phototaxis, it seems to be the same. In fact, when we first developed this adaptive model, we thought we were really clever and invented something new, only to discover that in the theory of chemotaxis in bacteria, it had been long well known. Right.
Sorry, I was sort of being fascinated by the earlier lectures, particularly. It's probably more of a biological question than a mathematical question, and also going back to your metronomes, when you were looking at the Volvox and the way it decides to specialize in terms of its equatorial region versus its polar region, and where it decides to actually flip, and that this is just some sort of random thing that it picks. Has anybody done some analysis whether this is somehow involved in the protein structure of the object? Because obviously the stuff we've done with amyloid plaques now, they seem to have a mind of their own, you know what I mean?
Well, so if I understand what you're asking, if we think about this inversion process that I was describing, it's not quite a random direction. There is a well-defined anterior-posterior pole, and the axis around which the inversion is occurring is pretty well, not perfectly well, pretty well aligned with that. So it raises the question of how do the cells at the equator know that they're the ones at the equator and they should do this? And the answer is we don't know. In other systems, the fruit fly, et cetera, there are gradients of chemical concentrations within a continuum of cells where each one is talking to the next.
Yeah.
But these are not talking to each other biochemically because they're separated by this extracellular matrix. So that's a mystery. It could be that it's actually programmed in via the age of the cells because there's a succession of division cycles that fills up this bowl to create this sphere, which then does its thing.
Yeah.
And in a sense, I think you can assign an age. It's not much different, but you can assign an age to each structure within it. So it's something like that, perhaps. And in fact, the real goal would be to do transcriptomics, where you look at the protein production at the single cell level in various places. Someone in my group is actually trying to do that with help from some biochemists.
It's just interesting because I asked Claude the answer and it refused to answer. So maybe somebody needs to look at it, which I've never actually
Sure.
Thanks, Ray. That was terrific. I have a short question and then more of a philosophical question. The short question is, when you had the experiment with the two lights and some went up and some went down rather than going through the middle, I thought the next thing you were going to say is, so we collected the ones up here and we collected the ones down here, and these had a high eye spot and these had a low eye spot. Is that too hard of an experiment to do?
So Shantanu has tried exactly that. It's a very hard experiment.
Okay.
Not a lot of data. The last I heard, it was sort of possibly working, and they're working on it. But that's the obvious experiment.
Okay.
Yeah.
And that my more philosophical one was, you sort of started the talk thinking about saying that we have coordination without communication. And I'm wondering can I think about this as something like Volvox that's figured out a cheat because it's rotating at a speed that's faster than it's turning, it's sampling what its neighbor is seeing. So even though it's not talking to its neighbor, it has some information about what its neighbor knows.
That's a fair statement. It's not, in a sense, an explicit part of the theoretical model, but it's a fair statement. It is sampling its environment by spinning, and the separation timescale seems to be a reasonable thing, but not always. And there, again, there's this singular perturbation with that short response scale. So it's actually, I still think something worth a lot of study is how much averaging is it doing? You know, it's a nice, you can develop a beautiful multiple timescale analysis with a solvability condition. We did it here to, you know, to get the equation in motion, but you get the same result as when you just wave your hands and say it. But it would be nice to really test this carefully. Yeah.
Maybe one last question.
So I think I'm thinking in particular about the two sources problem. So the kind of rays of light are emitted kind of parallel. So I'm thinking about difference between swimming towards a bright coast and swimming towards a lighthouse.
Absolutely, right. So this was chosen to be as if the light source is at infinity, like the sun. So it doesn't matter if the cells move or are distributed in a reasonable region within the central illumination, it's always at infinity. Actually, what NearGov was thinking about was organisms like birds or fish, you know, which could respond to a particular signal, and then as you move, its angular position is changing. So that would be another experiment to try, but we thought this was the simplest one because it removes the need to keep track of where you are. You always know the light is from infinity. Yeah.
Perhaps it could yield some curved trajectories, maybe, or
Say again? Yeah.
it could have some curved trajectories, maybe. Yeah. Okay. Thank you very much.
Yeah. Thank you very much, Ray, again for a wonderful set of lectures.
Thank you.